I'm very happy to be here and talk a bit about in the name, firstly, in the name of the Council of the Scientific Students Association of the Medical School of Szeged, to such a noble audience. So I was asked to perform this short introduction before the presentation of our students to give an overview of a very, very specific system which works in parallel and in association with the foundation. The goals are identical, and I would like to talk a bit about this system and the acronym, which is TDK in Hungary, or in the Hungarian higher education. So the history of TDK, or scientific circle, or scientific study groups of the Hungarian higher education goes back to the mist of time. Nevertheless, the roots probably can be found in a decision in 1951, when it was officially embedded into the higher education system of Hungary. So since that time, biannual congress is organized each second year in a kind of rotation in Hungarian medical schools. The last was organized by us last year, so in 2013. It was the 31st Congress. The system actually based on self-education. It's a voluntary-based, independent work of students and tutors. So both are, and I underline this word, volunteers working and doing practical research in leisure time as a hobby. And this way, the practical teaching, which by definition practical in this case, it reduces the time of raising or being a young scientist, so the learning curve is much shorter. And last but not least, it creates a bridge between theory and practice. It's a very, very strong connection. We've heard about the link between theory and practice, and we usually say in biomedical sciences that practice and theory are equal, or on the same level in theory, but in practice they are not. So I think that this system, which leads to the creation of young and talented group of new scientists each year, is probably part of the quality control, like the ubiquitin system of Hungarian higher education. And in this sense, the National Congress organized each second year is also a very important element of the strategic initiatives related to the quality of or quality control of Hungarian higher education. So looking back to more than 60 years of history, this conference series is clearly strengthening the relationship between students and tutors, or masters and apprentices, and it really gives nationwide recognition and publicity to us, to them. So until this moment I was talking about theory, and in the next few presentations we are talking and listening the presentations of our best student teams, so winners of the theoretical sessions of the last Congress, which was held in this March. So after this introduction, we are eager to listen to them. Thank you very much.
So thank you. My name is Hanna Fodor, and with my colleague Beáta Diósi, today we would like to present our latest results from the Master Research Group of the Institute of Biochemistry. Myostatin, a member of the TGF-beta superfamily, is mainly secreted from skeletal muscle, acting as an important negative regulator of muscle growth. Once reaching the target cells, myostatin binds to activin receptor type 2b, which in turn dimerizes with the activin receptor type 1. Due to receptor activation, the proliferation and differentiation of myoblasts will be inhibited through different intracellular pathways. Follistatin, a positive regulator of muscle growth, binds directly to myostatin, thereby inhibiting its activity. Another positive regulator is the insulin-like growth factor 1, which, by inducing ACT pathway, increases protein synthesis, decreases protein degradation, and enhances cell proliferation. The IGF-1 pathway can be inhibited by myostatin, which again results in the inhibition of muscle growth. Androgens, on the other hand, bind to androgen receptors and stimulate muscle growth by enhancing IGF-1 pathway while diminishing myostatin signaling. As a consequence, the enhanced production of myostatin leads to muscle mass decrease, for example, in cachectic patients, in muscle dystrophies, or in aging, whereas the lack of myostatin increases muscle mass tremendously. On the left-hand side, a myostatin knockout mouse is shown. Nevertheless, natural mutations of the myostatin gene have been described in several species, for example, in cattle and in human. Hypermuscularity is clearly seen in each case. The compact mutation is also a natural mutation of the myostatin gene, which has been fixed during long selection and inbreeding in the compact mouse line. The compact mutation does not affect the myostatin active domain, but its propeptide region, where a non-frame shift deletion of 12 base pairs occurs, as illustrated on this slide. Furthermore, modifier genes are also necessary for the full penetrance of the compact phenotype, but these genes have not been identified yet. In our earlier work, we have already performed a detailed morphological analysis of the compact muscle. Our present aim was to start exploring the molecular mechanisms leading to the hypermuscular compact phenotype. As a first step, we measured the transcript levels of myostatin and active receptor type 2B as part of the myostatin pathway, as well as messenger RNA levels of the positive regulators like follistatin, IGF-1, and androgen receptor. To ensure a reliable molecular analysis and to uncover the possible effects of the modifier genes, we have created a genetic control mouse line in which the myostatin gene was wild type, while the genetic background was identical with that of compact. Accordingly, if one measures differences between the wild type and the genetic control lines, these are likely to be the consequence of the modified genetic background of compact. On the other hand, if there is a difference between the genetic control and the compact line, it should be caused exclusively by the compact mutation of the myostatin gene. Generation of the genetic control mouse line was started in the end of 2010 by crossing wild type and compact mice. Heterozygous F1 generation, then we crossed individuals from this generation, and accordingly in the F2 generation we received wild type, heterozygous, and compact mutant mice, which were identified by genotyping as shown in the slide. The genetic background of these particular animals was about 25% identical with that of the compact line. During the first backcross, we selected F2 females with a wild-type myostatin and crossed them with compact males. Heterozygous B1 F1 mice produced again F2 generation, whose genetic background was already 56% identical with that of the compact. 4 more backcrossings were carried out, so we could increase step-by-step the ratio of the compact genetic background. Finally, in February 2014, we ended up with the B5 F2 generation, whose genetic background was 97% identical with that of the original compact line. These mice were then used as genetic control in our work. In our experiments, we analyzed 10 12-week-old compact, wild-type biopsy, and genetic control mice. Quadriceps femoris, gastrocnemius, and tibialis anterior muscles were removed from the mouse hind limbs. Their muscle mass were measured and then frozen in liquid nitrogen. Total RNA was isolated with 3 reagents from the tibialis anterior muscle, followed by cDNA synthesis. We analyzed transcript levels by qRT-PCR with the help of self-designed primers, as shown in the table, and normalized them to the housekeeping gene HPRT. The data obtained by the DDCT method were evaluated by one-way ANOVA using PRISM software. Our results are as follows. The body mass of the compact animals increased significantly compared to both biopsy and genetic control lines, but we detected a significant difference between wild type and genetic control line as well. The absolute muscle mass of compact animals increased significantly compared to both wild type and genetic control lines. By normalizing the muscle mass to body mass, the relative values of compact muscles were still significantly higher, revealing a disproportionate muscle mass increase. The difference between compact and its genetic control was even higher when compared to those of the biopsy in the most examined muscles. The messenger RNA level of myostatin increased significantly in compact animals compared to both types of controls, showing that the compact mutant myostatin gene is transcriptionally more active. The level of ActR2B messenger RNA decreased significantly in compact animals, but we detected similar low levels in its genetic control as well. Regarding follistatin, we detected a dramatic drop in compact animals as well as in their genetic control, but the compact muscles showed further decrease when compared to those of the genetic control. If we consider IGF-1, we could not detect any remarkable difference in transcript levels of compact versus control muscles. In contrast, androgen receptor transcript levels decreased compared to both types of controls, but it was significant only in comparison with biopsy mice. Summarizing our results, we should clearly discriminate between two situations, as it was mentioned before. In the first case, if one measures differences between biopsy and genetic control lines, these are likely to be the consequence of the modified genetic background of compacts. On the other hand, if there is a difference between genetic control and compact line, it should be caused exclusively by the compact mutation in the myostatin gene. Accordingly, the smaller part of the myostatin increase, the bigger part of the follistatin drop, and the decreased active receptor type 2B transcript levels are all consequences of this modified genetic background of compacts. However, this particular background did not have any effect on the transcript levels of androgen receptor and IGF-1. Altogether, these changes suggest that the genetic background of compacts enhances myostatin signaling. On the other hand, the bigger part of the myostatin increase, the further follistatin drop, and the decreased androgen receptor transcript levels are all consequences of the compact mutation in the myostatin gene, while neither ActR2B nor IGF-1 transcript levels were influenced by this particular mutation. These transcriptional alterations can be considered as compensatory mechanisms aiming to reverse the hypermuscular compact phenotype. We are planning further experiments to analyze the whole transcriptome and the changes in protein expression. Finally, I would like to thank to our tutors and to the whole staff of the Institute of Biochemistry. Thank you for your attention.